Hi, I'm Dr. Brian Kaufman, a retired family doctor and a CLL patient myself, and the co-founder and executive vice president and chief medical officer at the nonprofit CLL Society. Dr. Abdel-Wahab, can you introduce yourself, please?
Sure. Thank you, Dr. Kaufman. My name is Dr. Omar Abdel-Wahab. I am the chair of the Molecular Pharmacology Program at Memorial Sloan Kettering Cancer Center and a leukemia physician here as well.
And doctor, you treat a lot of patients, not just in CLL, but in other types of blood cancers. And one of the areas of research that you've been interested in is resistance. And one of your papers was quite complicated, caught my eye, and it talked about mechanisms of resistance in CLL. So, and this was specifically on the B cell receptor pathway. For just, quickly, why is the B cell receptor pathway an important target for anti-cancer drugs in CLL?
Yes, that's a really good question. So what we've learned over the years is that CLL cells are totally dependent on the B cell receptor for their survival. So drugs that can block the B cell receptor and B cell receptor signaling result in the death of CLL cells and basically can result in long-term remissions for CLL and CLL patients. And that's been the basis for the success of the drug ibrutinib, for example, as well as now the drugs acalabrutinib and zanubrutinib.
And these block at BTK, Bruton tyrosine kinase inhibitors, and they bind covalently or irreversibly. But resistance develops. Explain what that resistance is, because this is going to set up for some new areas of therapy that you've been researching. And so why don't you explain what that kind of resistance is, and then we'll talk about the newer BTK inhibitors.
Sure. Yeah, so currently there are 3 US FDA-approved inhibitors of this protein BTK. Those are ibrutinib, acalabrutinib, and zanubrutinib. And all 3 of those are irreversible or covalent inhibitors of BTK. And while they're incredibly successful and been really, really beneficial to hundreds, thousands of patients with CLL, they all have one problem, which is it is possible that people can develop resistance to those drugs because of a specific mutation that develops that blocks those drugs from interacting with BTK and blocking BTK. And so that has resulted in this need for other drugs that can overcome that resistance to these irreversible BTK inhibitors. And one example, yeah, go right ahead.
No, go ahead. Yeah, because I think you're going where I'm going is they develop new ways to bind that are reversible. Can you explain what that difference is and how that resistance, but now there's a resistance to those drugs. Yeah,
exactly. So now there's this new class, you could call them second or third generation of BTK inhibitors that work by different means. They block BTK reversibly, and they interact with BTK in a way that's unique compared to the FDA-approved BTK inhibitors, and they seem extremely promising. They're now in pretty advanced clinical trials. Several hundred CLL patients have received these drugs as single agent or in combination with other drugs. They seem safe and well tolerated. One of the drugs that's most advanced is called pirtobrutinib. And we're hopeful that eventually pirtobrutinib may become FDA approved and could be a really viable treatment if patients were to become resistant to one of the current approved FDA BTK inhibitors. Now, the problem is that with any form of therapy in cancer, eventually patients could become developing resistance against them. And so we're now starting to identify that these newer class of BTK inhibitors that I just talked about can also experience resistance in a unique way. And that's what's resulted in this paper that caught your eye, which is we've now begun to think about new ways to inhibit the B cell receptor signaling pathway in BTK that's completely unique from just these BTK inhibitors that we've talked about to date.
One of the concepts that really needs some hand-holding here for me, and I think for our listeners, is that you discovered that even when the enzyme, because a kinase is just an enzyme, and an enzyme is just something that makes the chemical reaction happen, but even when that's blocked, you discovered that the activity remained. And so this is the term that I think you're using is kinase dead. You talked about this concept of scaffolding. I know this is a complicated situation here to explain, but can you put this in patient-friendly terms so we can understand what issue you're trying to overcome? Because it's not just the mutation of the binding site. There's more. That doesn't explain. There's a lot of resistance to BTK inhibitors that's not related. People progress even when they don't have a mutation at the binding site.
Right. Yeah, this is a technical point, but I'll explain it. So BTK, Bruton's tyrosine kinase, that's what the acronym of BTK stands for, is an enzyme that is a protein that has this activity in the cell. And its enzymatic activity is thought to be what drives CLL cells to grow and proliferate and create clinical problems. And what we found is that when patients become resistant to ibrutinib or some of these other BTK inhibitors, the enzyme no longer functions, but still has some new activity that allows CLL cells to survive. And that has a practical importance because what we've figured out is that this new mutant form of BTK, even though it can't signal and function the way a normal BTK enzyme can function, it takes on some new activities and stimulates other signaling proteins in the cell that now that we've discovered this, themselves become new therapeutic targets for other drugs that could be really exciting new drugs for CLL. So that's why it's got some practical importance even just beyond studying this at a scientific level.
There's another enzyme that we've heard less about, but we see it in papers called PCL gamma 2. And that's what I understand is downstream. Downstream means the signaling happens at the surface of the cell, and in the nucleus is where all the activity, the DNA is that sends the messages to proliferate, stay alive, and BTK is in between that, and between BTK and the nucleus is PCL gamma 2, and that's what makes it downstream. Explain what PCL gamma 2 does and how your research relates to that. This is a particular interest to me because I have a PCL gamma 2 mutation. Yeah.
Right. Yeah, so PLC gamma 2 is indeed an enzyme, just like BTK. It's a little bit different in its activity, but it is immediately downstream of BTK. So as you said, at the outside of the cell is the B cell receptor, and that is thought to be always active in CLL cells abnormally, and that's what drives the growth of CLL cells and the clinical problems in CLL. So the B cell receptor eventually signals to BTK, and then BTK in kind of a tag team will signal PLC gamma 2, and then that will stimulate the growth and survival of CLL cells. And so these drugs like ibrutinib, acalabrutinib, and zanubrutinib stop that signaling by blocking the ability of BTK to activate PLC gamma 2. And then, so one other means of resistance to these drugs is that PLC gamma 2 can become mutated and activated so that it doesn't rely on BTK anymore. And that is an important clinical problem that individuals, even such as yourself with CLL, have faced at times.
So it sounds like blocking the enzyme at BTK isn't the whole story, and we know that because we're seeing resistance, and we see resistance for the non-covalently binding drugs like pirtobrutinib. You're looking at some new class of drug that might overcome these problems. Can you explain what these are and why you're excited about this, why you're doing research here?
Yes, absolutely. So as we've just been talking about, the current FDA-approved drugs that inhibit BTK, they block the enzymatic function of BTK. So BTK, the protein is still there, it just can't function anymore. But now we were discovering that even when the enzymatic activity of BTK is blocked, that may still allow the cells to signal and no longer care that ibrutinib or any other BTK inhibitors exist. So that has led us, along with a number of companies, to explore a new therapeutic area which are drugs that degrade or erase the BTK protein altogether. So we eliminate the protein from the cell, and then that is a new way, we think, to treat CLL that looks really, really promising. So this is now a third category of drugs that target BTK and CLL. There are a few of these drugs that are in clinical trials currently. They are all in what we call phase 1 clinical trials, so the very earliest stages of testing in patients. They're given as single agents to try to make sure that they're safe, and to see what kind of clinical activity they might have and what doses are ideal. And so we think that this is going to be really exciting because we found that these drugs can also, some of them can degrade the mutant forms of BTK as well. So they could potentially overcome resistance not only to ibrutinib, acalabrutinib, zanubrutinib, but to pirtobrutinib as well. So they look really exciting if they can be given safely and have clinical activity, which is what's being investigated in early clinical trials right now.
Yeah, and we have an interview on this topic. We interviewed Dr. Wiesner about this in the NIH. I think did some of the early research on that. And there was a paper presented that presented some early promising clinical data. Even, I think there was just maybe just a very small number of patients on this, but it was certainly encouraging.
Correct. Yeah, it's really early days, but there are at least 3 different BTK degrader drugs that are in clinical trials now. So I think we're going to learn very quickly, is this a, exciting therapeutic avenue, and does it have great potential for patients or not? We'll see. But certainly scientifically, it seems to have really great rationale.
Would this help overcome the issues that we just talked about? And am I right in understanding this? If you eliminate the enzyme completely, then any kind of changes the enzyme makes after it's blocked the BTK would also be eliminated. Is that
correct?
Correct. And any scaffolding functionings would be eliminated.
Correct. That's the thought and the rationale behind it, and why we're really excited about it. It is new, this whole modality of what they call targeted protein degradation, of drugs that will go and erase certain cancer-causing proteins from the body, is a really, really new concept, and we don't yet know how effective they're going to be in patients. They're being tested for all forms of cancer, but also in this area of BTK degraders. So it's a very exciting area. We'll have to see where it goes.
Dr. Abdel-Wahab, any final thoughts that you want to share with patients about this kind of research? Are there trials that they can be looking for and enrolling in?
Sure. Yeah. One thing is that there are, as I mentioned, a number of clinical trials of this new type of BTK degrader drugs that are ongoing throughout the world. So it's not just the United States. So patients even outside the United States, in Asia and Europe, Middle East, South America, may be able to get access to these therapies if they're in need. They're generally for patients that have relapsed 2 or more lines of treatment. And so that's something to keep in mind. So one could go to clinicaltrials.gov, for example, and just type in BTK degrader, and some of these will pop up. We could probably provide some names of these drugs on the website if that were,
I'll put in some links for that.
Yeah, but there are several of these clinical trials from multiple companies throughout the world ongoing, Australia, Asia, et cetera. So hopefully people could get access to these, if they were eligible for those studies.
Well, Doctor, I thank you so much for trying to simplify a very complicated concept. And I'm so grateful for the research that's been going on to look at new ways, because it seems like we block one pathway and the cancer finds a way around that. And this seems like it's a more thorough way of blocking that pathway, and hopefully a way that makes it a little harder for the cancer to figure out a way around it. So I'm very grateful for the research you and your colleagues are doing, and I look forward to hearing more about this in the future. Thanks so much.
Thank you. Thanks for the opportunity to talk about this.
Thanks.